Even at a microscopic level, there is a bit of selective dating going on. While we're often taught that fertilization is a frantic race where the fastest sperm wins, modern science shows it's actually more complex than that. Instead of pure speed and chance, the process involves interaction and selection. The human egg isn't just a passive waiting cell. Research shows it can influence which sperm succeed. When sperm enter the reproductive system, they begin a long and challenging journey. Out of millions released, and only a few thousand make it as far as the fallopian tubes. On the other hand, after ovulation, the egg travels into a specific meeting point in the fallopian tube called the ampulla. This is where fertilization most often happens. The egg's survival window is short, usually about 12 to 24 hours. As it arrives there, the egg and its surrounding cells begin releasing signals that indicate it's ready for fertilization. For sperm, timing is critical. They have to be in the right place at the right moment to respond to these signals and reach the egg. As sperm get closer, the egg and its surrounding cells release chemical signals called chemoattractants, almost like a scent trail guiding them. These signals can attract some sperm while potentially ignoring others. The egg is essentially sending a GPS signal that only certain sperm are tuned into. While a sperm might be a marathon winner in terms of speed, the egg is looking for something much deeper: genetic compatibility. Actually, the egg doesn't want the best sperm in a general sense. It wants the sperm that best complements its own DNA. Here is how the choice actually works. The chemicals released by the eggs don't attract all sperm equally. An egg's scent may be more attractive to the sperm of one individual over another. Studies suggest this preference may be linked to genetic differences, especially in immune system genes known as MHC. Greater diversity here is often associated with stronger immune protection in future offspring. When a sperm reaches the egg, the process becomes a coordinated chemical and mechanical interaction. As it encounters the zona pellucida, the egg's outer layer, the sperm undergoes the acrosome reaction, releasing enzymes and using strong tail movements to help penetrate this barrier. However, passing the outer layer does not guarantee fertilization. Final fusion occurs at the egg's membrane and depends on specific molecular binding between the sperm and egg. But when chosen sperm successfully fuses with the egg's membrane, the egg undergoes a massive chemical shift that instantly hardens the outer shell, locking out all other contenders forever. This selective process is the reason why fertilization is such a miracle of biology. It isn't just about who is fastest, it's about which sperm carries the genetic code that best complements the egg's own DNA. So if the egg is so picky and the process is so selective, why do things still go wrong? The truth is that while the egg is an incredible gatekeeper, it isn't perfect. It is a biological system, and biological systems have limits. Actually, the egg can only screen external cues. So a sperm might be a strong swimmer and interact correctly with the egg, yet still carry hidden genetic changes or recessive disease genes. Those aren't always detectable at the moment of fertilization, which is why biological selection, while remarkable, isn't foolproof. Also, sometimes the sperm and egg are both perfectly healthy when they meet. But a glitch happens during the actual merging or the very first cell divisions. Evolution doesn't aim for perfect, it aims for good enough to survive. The egg's choice significantly reduces the risk of many problems, but it can't eliminate them entirely. Actually, if the egg were too picky, Fertilization might never happen at all.